So we talked about the structure, this whole thing, how we separate the strands via helicase, how the single-stranded binding proteins hold the two parent strands apart, how topoisomerases keep them from getting tangled. So now let's talk about how we actually make new DNA strands. So basically, we want to add DNA nucleotides that match these. So let's say this was CAT. We would add GTC right there. So that's what we're trying to do, to build a complementary strand. So it'll be identical to this strand, but attached to this strand. So that's what we're trying to do. But the problem is that, well, actually, the enzyme that adds DNA molecules, I wish there were a better picture of this, is called DNA polymerase. And there's several DNA polymerases. In bacteria, we talk about 2, I think. There's like 11 in eukaryotic cells, but we're just going to mostly refer to them all as just regular DNA polymerase. They add on the nucleotides. However, they can't start a nucleotide strand. They need to add on to an already existing nucleotide strand. So they just can't come here, find CTGC, and start adding. They need to have something there already existing, which is a problem because there's nothing there, right? We're starting from nothing. So what we do is there's a molecule called primase. Right? We need a primer. We need molecules already there. That way DNA polymerase can come on and attach them. And so what we do is we form this primer by an enzyme called primase, which puts some nucleotides onto the strand. That way we have a place to start. However, primase does not work with DNA, it works with RNA. And so our primer, where the DNA replication is going to start from, right there, where we're going to start adding nucleotides that are complementary to this strand, is actually made of RNA, not DNA. Because primase adds RNA, not DNA, for whatever reason. And so primase starts by adding just a few nucleotides, one at a time, that are complementary to the DNA strand. And the primer is usually about 5 to 10 nucleotides long, and it's complementary to the parent DNA strand. And then what happens is the new DNA strand is going to start growing from the 3' end of the molecule, always. Okay, so once we have our primer in, our molecule is called DNA polymerase, and that's going to continuously add DNA nucleotides to our ever-growing strand to make a new strand that is complementary to the parent strand. And the rate of DNA elongation is about 500 nucleotides per second in bacteria and 50 per second in human cells. So that's pretty fast. First of all, in bacteria, 500 nucleotides a second. 500 nucleotides. In humans, still 50 nucleotides. That's huge. Okay, so we need to talk about the antiparallel nature of the double helix. That's right there. So we talked about this a little bit last time, but basically DNA is made up of 2 strands. You know that. They're like, and they have directionality, like a one-way street. And so they both run in the same direction. For example, on a one-way street, there's no one-way streets around here that I can think of. On a one-way street, you can only turn one direction, right? If the one-way street is going this way, you can only turn right. You turn left, you're not in very good shape. It only goes one way. It's the same thing with DNA molecules, except instead of driving one way, we have a 5' end and a 3' end. And remember that the primes, this picture, refer to the, I'm going to jump way back. The primes refer to the carbons on the sugar. So basically, here's the 5-carbon sugar that's part of the sugar phosphate backbone of our DNA molecule. This carbon, we count carbons, so this would be 1, 2, 3, 4, 5. This is 3 right here. This is 5. So this is the 3' end of the carbon. It's the 5' end of the carbon, of the sugar. So this is the 3' end of this DNA molecule. This is the 5' end of this DNA molecule. So it has directionality. It goes from one end to the other. 3' to 5'. This one also goes from 3' to 5', except I'm covering the 5'. Except, you might notice that they are going in opposite directions because they run anti-parallel to each other. So it's like 2 one-way streets that are running in reverse to each other, like a divided highway. Like how on Rosecrans, except when you get to a stoplight, you can't get to the other side. right? They're divided. One goes this way, one goes this way. They're anti-parallel to each other. And so that's really important when it comes to DNA replication, and we shall see why. Okay, the reason for that is because DNA polymerase can only add on nucleotides to the 3' end of the DNA molecule. For whatever reason, the chemistry of the molecule, it only can add on DNA to the 3' end of a growing strand. So that means that nothing's happening at the other end. So it can only go one direction. So it can only elongate from 5' to 3'. I'm confusing myself. Okay, it can only add on to the 3' end, so we're going to keep adding on, so it's going to be 5' to 3'. So basically, here we have it. This is the 5' end of the molecule. Here's the 3' end. DNA polymerase can only add on to this end right here. So it can only go this way. So this end is, nothing's happening there. At the origin of replication, it's only going 5' to 3'. That's all it's doing. So that nothing is happening over here. So that's what causes the problem. And basically, along one of the template strands, DNA replication is going to happen for forever. So here's one of the template strands right here. Along this one, DNA polymerase picks up where the RNA primer was and just takes off and it just goes and it moves into the replication fork and it's going to keep going until it hits the next replication bubble. It's continuous, no problem, no pasa nada, it's just going. Because it's adding on at the 3' end and that's fine. However, the other strand, okay, so here we go. Here's the origin replication, picks this strand, it goes, until eventually comes in over here and meets the other end. But the other molecule, the other one down here, these pictures are not very good. Right here, how do I explain this? These pictures are not doing a good job. So right here, there's no primer. So basically what happens is we have a leading strand. That's the one I was just talking about. It goes. It's kind of leading its way into the replication fork, and that's why it's called the leading strand. It's taking off and going into the replication fork. It also is happening over here at this end of the replication bubble, except it's happening in the other direction because this strand is now, instead of being 5' to 3', now this one is 5' to 3' the other direction. So it's going the opposite direction, the leading strand, and this one's leading into the replication fork. And so basically what happens is as the replication fork starts to open slowly and slowly, so right here, see the leading strand is only this far. As it moves, it gets all the way down here, okay? So it's opening and opening and opening the parent molecules little by little. So what happens is, as they open, so at first, it's only open to right here. Then it's open to right here. Now it's all the way open to right here. In a minute, it'll be open to right here. So it's slowly opening. But nothing is happening on this strand. So what happens is, we just do these, the other side, we do them in little pieces. And we call those the lagging strands. So it picks up when it's far enough, and it goes. Then it picks up again when it's far enough, and it goes. Picks up again when it's far enough, and it goes. So instead of doing it in one continuous motion, it does it in little tiny pieces. I have a good picture of this. Okay, so this is called the lagging strand. And it's not lagging because it is slow. It's lagging because it is doing it little by little. So the leading strand goes all the way in, and the lagging strand is catching up. And each of these little sections of the lagging strand, one right here, one right here, one right here, they're called Okazaki fragments. So basically, let's look at what is happening right here. So right here, this side is the leading strand. We knew that that was happening. This is on the other side of the replication fork, on the other strand. So it opens up and an RNA primer gets put on. Then, by primase. Then DNA polymerase comes and it adds the DNA nucleotides like it would. Then it jumps back a little further because now the replication fork is open a little bit further so it can. Jumps back, finds another primer, put there by primase, and it goes again. And you'll see that this Okazaki fragment eventually catches up to this primer. So that's what's happening right here. There's one Okazaki fragment, another Okazaki fragment, another Okazaki fragment, and they're all started with primers, RNA primers. And so the Okazaki fragments are going. They catch up with the primer, and it's done. So it jumps back again and does it again. The DNA polymerase does. And so now we have this kind of continuous piece of nucleotides But some are RNA, some are DNA, some are RNA, some are DNA. And obviously we want them all to be DNA because that's what we're making, DNA. And so what happens is there's a molecule called ligase, and it comes and it cuts out the RNA nucleotides and it replaces them with DNA nucleotides. It gets rid of the primers and puts DNA in there. And that's its job. And so we can see here what this arrow here means is that replication is moving in this direction. So even though replication has to happen this way because of the directionality of DNA, the direction of replication is still going this way, which is why we keep jumping backward and jumping backward and jumping backward. This is what we just saw. Okay, so here's a better picture. The leading strand is going continuously like it should. But this is opening slowly and slowly and slowly, and so here it just keeps putting new primers on the lagging strand and starting again. Putting new primers on the lagging strand and starting again. And so DNA polymerase 3 is the one that adds nucleotides until it reaches another primer and then it stops. And then DNA polymerase 1 comes and cuts out the RNA nucleotides and puts in DNA nucleotides. And you can't see it, can you? Oh, that didn't work. And ligase cuts out and fixes them together. So as the replication fork gets open further and further, because this DNA polymerase 3 continues to open it, there will be another RNA primer probably right here, and then DNA polymerase will come in, make another Okazaki fragment, and it'll continue and continue and continue. This is going to keep happening until two replication forks hit each other. Because remember, there's so many origins of replication, so there's a ton of replication forks. So that's basically what's happening over and over and over again.